Hello everyone, welcome to MMBS111 Introductory Microbiology. This mini lecture is going to cover content from section 7.1, where we're going to be discussing metabolism and the role of enzymes in metabolism. There are 6 learning outcomes for this mini lecture. First, describe the relationship among metabolism, catabolism, and anabolism. 2, fully describe the structure and function of enzymes. 3, describe the relationship between enzyme names and their function. 4, differentiate between constitutive and regulated enzymes. 5, diagram 4 major patterns of metabolism. And finally, explain the processes of enzyme inhibition, non-competitive inhibition, repression, and induction. So we're going to begin by talking about metabolism. In science, metabolism is defined as all chemical reactions and physical workings of the cell. So metabolism includes every single chemical reaction that occurs within cells. And there are 2 types of metabolism, anabolism and catabolism. And anabolism is all of the chemical reactions that involve building large complex molecules from smaller substances or smaller molecules. And anabolic reactions always require energy input in order for that to happen. Catabolism is the opposite. Catabolism is the breakdown of large complex molecules into smaller molecules, and it results in the release of energy when the bonds in those complex molecules are broken. So these reactions don't happen without a little bit of help, and that help comes in the form of enzymes. Enzymes are reaction catalysts, and a catalyst is a substance that increases the rate of a chemical reaction. It means it helps that reaction happen more quickly. And with the input of less energy, as you can see in the graph on the left, uncatalyzed reactions without the assistance of an enzyme require more energy than reactions that are catalyzed with the help of an enzyme. Enzymes do not become part of the reaction, so they sort of stimulate the action. They reduce the energy that is required, but they are not considered part of the reactants or the products. They don't get used up in the reaction, so they're not consumed. And they also do not create a reaction. So these reactions would happen without the enzyme, but they happen more efficiently with the enzymes and more quickly. So let's look at how enzymes work. Enzymes serve as a physical place for reactions to occur. This is where substrates get sort of positioned for certain reactions to happen. So enzymes are always larger than the substrate that is involved. And they present a very unique active site, and that active site is unique for the substrate. So we don't have enzymes that a random substrate just binds to and things happen. Every enzyme is specific to the substrate or the reactants that are involved in the reaction. The enzyme participates directly in producing changes in the substrate, but it doesn't get used, as I said before, in the reaction. And once the substrate is changed and the products are produced, the enzyme is then free to bond to a new substrate. So they don't get used up. They can repeat this process multiple times within the cell. You can see in the model on the right how this works. So we have an enzyme with an active site. That initial substrate is going to bond specifically to that active site. And that active site has a very unique shape that fits only that substrate. When the reaction occurs and we have this bonding happen, we get an enzyme substrate complex. We looked at this before in previous chapters. Once the substrate bonds, stress on that bond occurs at the active site, and that stress breaks the bond, and then those products are released. So in the case of sucrose, when we're trying to break this down into its component parts, glucose and fructose, the bond that holds the glucose and fructose together becomes stressed as it has bound to the active site in the enzyme. And that stress breaks the bond, and then those two smaller molecules are then released. So we have some different structures and sort of combinations of enzymes that we're going to look at here. A holoenzyme is a conjugated enzyme that includes one or more cofactors, and those cofactors can be metal ions, they can be coenzymes, or they could be a combination of both of those things. So the apoenzyme is just the protein itself, and that's the large structure that comprises the majority of the enzyme. And then we have these other cofactors that bind to that apoenzyme that help with the functionality of the holoenzyme itself, which is the combination of both of those. And as I mentioned before, the enzyme substrate union, or that complex, is very specific. So you can see in the bottom right there, we have two different substrates, but only one can bond to that enzyme because it fits the active site, whereas the other one does not fit, so it will not bond to that enzyme. And that's important to know because enzymes, this reaction is not random, but it's very specific between the enzyme and the substrate. So let's look at coenzymes and cofactors here for just a minute. A cofactor is a substance that helps to activate an enzyme. Cofactors are used because they can help bring the active site and the substrate closer together. Examples of cofactors include iron, copper, magnesium, zinc, any number of metallic ions. Coenzymes are a type of cofactor, and they carry and transfer chemical groups from one substrate molecule to another. These are often organic compounds that are derived from vitamins. And they can work in conjunction with a metallic cofactor. So you can have enzymes that possess one or both of these compounds. There are 6 general classes of enzymes, and each of these enzymes performs a specific function, and it's sometimes stated as part of the name of the enzyme. So we have oxidoreductases, and these are enzymes that transfer electrons from one substrate to another. Dehydrogenases transfer a hydrogen from one compound to another, but they are included in this group of oxidoreductases. We also have transferases that transfer functional groups from one substrate to another. Hydrolases, these cleave bonds on molecules with the addition of water. So water is a component in the reaction and is necessary in order for those bonds to break. We have lysases. These are enzymes that add groups to or remove groups from double bonded substrates. Isomerases are enzymes that change a substrate into its isomeric form. And finally, ligases. These enzymes catalyze the formation of bonds with the input of ATP and the removal of water. So enzymes are necessary for many reactions within the cell, but enzyme action needs to be regulated, and there are a number of ways that this action is regulated within cells. Constitutive enzymes, as you see in the diagram on the right, these are enzymes that are always present within the cell in a constant amount. So the amount of that enzyme does not change within the cell, no matter what happens with the substrate. So if we increase substrate, we do not increase the amount of enzyme. That enzyme amount remains constant, okay? And these enzymes are typically essential metabolic enzymes that are necessary and need to be present at constant amounts at all times in the cell. Then we have regulated enzymes, and these enzymes vary depending on the amount of substrate within the cell. So if we increase the amount of substrate, this induces the cell to produce more of that enzyme. If the amount of substrate is reduced in the cell, then the cell will not produce enzymes. The activity of enzymes is really dependent on the environment within the cell. Enzymes are proteins, and they are sensitive to things like temperature, pH, osmotic pressure. So changes in those characteristics within the cell also affect the action of enzymes. And that can lead to instability of the enzymes and eventually denaturation. And we talked about denaturation in chapter 1 when we discussed proteins. Denaturation inactivates proteins. These proteins lose their native shape. Bonds are broken, and it prevents the normal substrate from attaching to the active site of those enzymes. We have several patterns of metabolism that occur within cells. Metabolism is a series of linked chemical reactions, and these reactions are all catalyzed by enzymes. There are 4 general patterns of reactions that we see within cells, as you can see there on the right. The first is a linear pattern, and we have one reaction followed sequentially by the next several reactions. An example of this is glycolysis, and we'll look at glycolysis in this chapter in a little bit later sections. We also have cyclic patterns where a compound enters the cycle. We have several inputs and outputs in the cycle, but this process continues in a cyclical pattern. And then finally we have branched patterns of metabolism, where we might have one compound, and through a reaction, that reaction may branch into several other reactions, or we might converge 2 separate chains into one. Amino acid synthesis is an example of a branched pattern of metabolism. There are a few controls within cells that directly affect the action of enzymes, and these are competitive and non-competitive inhibition. We can inhibit or prevent or control the action of enzymes with different substrates. So in the first example, competitive inhibition, the enzyme activity is inhibited by a competitive molecule. So this competitive inhibitor has the same or similar shape to the normal enzyme substrate, both molecules compete for a place on the active site of the enzyme. So if the competitive inhibitor bonds to the active site of the enzyme, it then prevents the action of the enzyme working on the normal substrates. So that reaction is blocked because no products are able to be produced from the normal substrate. There is also non-competitive inhibition, where an enzyme might have two bonding sites, an active site and a regulatory site. And as long as the regulatory site is free, the normal substrate can bond to the active site and the reaction proceeds as normal. However, if the regulatory molecule bonds to the regulatory site, this prevents the normal substrate from bonding to the active site, because the bonding of that regulatory molecule on the regulatory site changes the shape of the active site, and the normal substrate no longer fits the active site, so the reaction is blocked. We can also repress enzyme production genetically, and that's what you see here in this model. DNA is responsible for controlling really all of the activities that happen in the cell. And if, for instance, we have too much product being produced within the cell, then the excess can actually bond to the DNA, that gene that codes for that enzyme production. And then the RNA no longer gets produced and translated into a protein. Proteins don't get made, and so the enzyme is no longer produced. When the product level from that enzyme decreases, then that product unbonds to that section of the genetic material, and then RNA or DNA can be transcribed again into RNA, and RNA then will form the proteins, and that cycle will continue. So this is enzyme repression. Another way that we can control enzyme synthesis itself is through induction. And this is dependent on if substrate is present in the cell for that enzyme. So if there is substrate within the cell, then the genetic material is induced to produce the enzyme needed for that substrate. If the substrate is absent, then enzyme production stops. So here's an example of induction in E. coli. So if we grow a strain of E. coli in the lab, and it is inoculated on a medium that contains lactose only, that E. coli will produce the enzyme lactase to break down the lactose. If we grow a strain of E. coli in the lab that's inoculated on a medium that only contains sucrose, it will stop producing lactase, and it will begin producing sucrase. So this kind of control allows the organism to produce only what is needed. So it prevents the organism from wasting energy for making enzymes that are not necessary because the substrate is not present within the cell. All right, so that's the end of section 7.1. Make sure that you go through these questions, that you can answer them correctly, and go back to the learning objectives. Make sure that you understand each objective. Remember, you can watch the videos multiple times in order to ensure your understanding. One more section that I've added based on the test is critical thinking. So this question or this scenario states PABA is a metabolic intermediate of folic acid formation. Sulfanilamide is an antibiotic that interferes with the production of folic acid in bacteria. Based on the molecular structure of these two compounds and the description of their functions, how does sulfanilamide interfere with folic acid production? In order to answer this question, you need to be able to understand the regulation of enzymes and how enzymes can be controlled and regulated within the cell. So I would encourage you to go back to the section that discusses enzyme regulation and see if you can identify a process for enzyme regulation that might fit this scenario. Remember, if you have any questions about any of the content, please send me an email or ask me in class. And I hope that this video was helpful.